discussion, I'll be talking about the citric acid cycle. First of all, what is a citric acid cycle? It is a key metabolic pathway that connects carbohydrate, fat, and protein metabolism. The reaction of the cycle carried out by 8 enzymes that completely oxidize into acetate, a 2-carbon molecule in the form of acetyl-CoA, into 2 carbon molecules each of carbon dioxide and water. But what are the events happen inside the citric acid cycle? Let's find out.
We know by now that glycolysis takes place in the cytoplasm and leads to formation of 2 molecules of pyruvate per molecules of glucose. These pyruvate molecules are converted into acetyl coenzyme A, also known as acetyl-CoA, and this happens inside the mitochondrial matrix. Therefore, acetyl-CoA is the key metabolic intermediate that links glycolysis and the citric acid cycle. For clarity, I will not show the coenzyme portion of acetyl-CoA and instead are simply representing it as 2-carbon molecule acetate. As I said earlier, the pyruvate gets into the mitochondrial matrix depend on a carrier protein that transports pyruvate into the mitochondrial matrix, where there it is oxidized to form acetyl-CoA by the enzyme pyruvate dehydrogenase. In this reaction, a molecule of carbon dioxide is generated as pyruvate is oxidized to acetyl-CoA and a molecule of NAD+ is reduced to NADH. The citric acid cycle consists of the following 8 steps and also harvests chemical energy producing a total of 3 NADH molecules. In the first step of the citric acid cycle, the enzyme citric synthase combines the 2-carbon acetyl group from acetyl-CoA with a 4-carbon molecule oxalicetate. This generates the 6-carbon molecule citrate. In the second step, citrate is isomerized into isocitrate by iconitase. In the third step, isocitrate is oxidized to isocitrate dehydrogenase, generating 5-carbon molecule alpha-ketoglutarate. In the process, 1 carbon dioxide molecule is released and 1 NAD+ molecule is reduced to NADH. The fourth step, alpha-ketoglutarate, is decarboxylated and oxidized to produce another molecule of carbon dioxide by alpha-ketoglutarate dehydrogenase. The resulting molecule is combined with coenzyme A, forming succinyl-CoA. In addition, another molecule of NAD+ is reduced to form NADH. Note that in this diagram, I am showing the product of this reaction as succinate and are omitting the coenzyme A for clarity. In the fifth step, succinyl coenzyme A is converted into succinate by succinyl-CoA synthetase, releasing coenzyme A. In this reaction also produces 1 molecule of 1-GTP, another high-energy molecule which is omitted from this diagram. In the sixth step, succinate is oxidized to fumarate by succinate dehydrogenase. Unlike all the other enzymes in the citric acid cycle, succinate dehydrogenase is embedded in the inner mitochondrial membrane and also a component of the electron transport chain in which it is called complex 2. As complex 2 oxidizes succinate, it receives 2 electrons that will ultimately pass on to coenzyme Q, in which turn will deliver its electrons to complex 3. Another interesting aspect of complex 2 is that it uses FAD rather than NAD+ to accept electrons. In the seventh step, fumarate is converted to malate by the enzyme fumarase. And finally, in the 8th step, the enzyme malate dehydrogenase oxidizes malate to reform oxalicetate and reduce a molecule of NAD+ to NADH in a process. This regenerated oxalicetate can then combine with another acetyl-CoA in the next cycle.
Thank you for watching. I hope you learned something from this video. This is Eliza R. Video Peranya signing off.